Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Imagine being able to listen in on the unique conversations happening inside thousands of individual cells, not just, a blurry average signal.
Right.
What if you could do that for just pennies per cell and way faster than anyone ever thought possible? Today, we're diving into a really groundbreaking paper that completely transformed how we map the intricate world of biology. It basically took single-cell analysis from this niche, really expensive thing
Yeah, super laborious.
to an industrial-scale powerhouse.
Absolutely. Let's say before 2015, trying to figure out gene expression in individual cells was a monumental challenge.
Right. What were the main hurdles?
Think about the old methods like microarrays. Okay, they could measure lots of genes, sure, but the picture was so blurred.
An average, basically.
Exactly. You lost crucial details like splice variants, different versions of the same gene transcript. Gone. You just got this mush of maybe 1 million different cell types.
Which hides all the interesting stuff, the rare cells.
Completely. And then the single-stem methods that did exist, usually plate-based sequencing, well, they could maybe handle a few hundred cells.
And cost a fortune, I bet.
Oh, exorbitant costs, immense amounts of lab work. The real bottleneck was just that every single cell needed its own separate reaction.
Ah, okay.
Scaling that up, impossible. So complex tissues like the brain or the retina with all their different, sometimes rare cell types, they were pretty much a black box if you only did bulk analysis.
You just couldn't see the individual players.
Nope. Couldn't resolve them or figure out their specific roles.
Okay, so that bottleneck was really holding things back for bigger studies. It sounds like what was desperately needed was a way to, tag each transcript from each cell reliably.
Yes.
And do it for thousands, maybe tens of thousands of cells without spending millions of dollars.
Precisely.
And this wasn't just, academic curiosity, right? For functional genomics, precision medicine, getting that cell-by-cell view was critical.
It really was.
So when the authors were thinking about this, what was their big idea? Their, core concept to smash through that wall?
Their hypothesis was, it was pretty bold. What if tiny droplets, engineered just right, could act as individual reactors, like nanoliter-sized test tubes?
Okay.
And crucially, what if each one carried a unique molecular name tag? That's where the genius of Drop-seq really started.
Name tag and a droplet.
Yeah. The mission became build a platform to process over 10,000 cells in one go, count the transcripts digitally, and slash the cost down to just cents per cell.
Wow, okay, so how did they actually do that? Giving each cell its own tag without physically separating them sounds like the magic trick.
It really was the core innovation. It revolved around these tiny things called microparticles, or beads. Think of them as tiny delivery systems for special DNA primers.
Okay, beads with primers.
Right. And to make those primers unique for each bead, they came up with this super clever method called split and pool synthesis.
Split and pool. How does that work?
Imagine you have millions of these beads in one big pool. You split them into 4 groups.
Okay.
To each group, you add a different DNA base, an A, T, C, or G. This adds one unique letter to a barcode that's growing on all the primers on those beads.
Nice.
Then you pull them all back together, mix them up, and split them again into 4 groups. Add another base. Repeat.
How many times?
Just 12 cycles. Seems simple, right? But doing that 12 times created, wait for it, 16.8 million distinct 12-base pair cell barcodes.
Whoa! 16.8 million unique tags.
Exactly. And here's the key insight. All the primers on one bead have the same barcode, but each bead gets a different one. It's like giving every potential cell its own microscopic zip code.
Okay, that's brilliant for the cell ID, the zip code. But just knowing the cell isn't enough if you want to count how many copies of each gene message there are, right?
Yeah.
You need more precision.
You are absolutely right. That's the next piece. To count individual RNA molecules digitally and, crucially, to avoid overcounting because of PCR amplification, where one molecule gets copied many times.
Yeah.
Yeah, the PCR duplicates problem.
Exactly. They added something called unique molecular identifiers, or UMIs.
UMIs, okay.
After making the cell barcodes, they did 8 more rounds of synthesis, but this time it was degenerate synthesis.
Degenerate.
Meaning, instead of adding just one specific base, they randomly added any of the 4 bases at each of those 8 positions. This created 65,536 unique 8-base name tags for individual RNA molecules.
Wow, so a tag for the cell and a tag for the molecule itself.
Precisely. So each primer on the bead ended up with 4 key parts: a PCR handle bit, the unique cell barcode, the unique molecular identifier, and then an oligo-DT sequence.
The poly-A catcher.
Right, that string of T's that grabs onto the messenger RNA molecule specifically.
That is incredibly elegant. So you've got these super smart beads, but how did they get them efficiently paired up with single cells inside those tiny droplets? That sounds like another major hurdle.
That was the microfluidics part. They designed this specialized co-flow device. Picture 2 streams of liquid flowing side by side.
One stream has the cell suspended in it. The other has the barcoded beads, also suspended in a lysis buffer, something to break the cells open later.
Got it. Cells here, beads there.
These 2 streams then meet a stream of oil right at a specific junction. And the physics of the flow, the engineering of that junction, it's just beautiful. It creates over 100,000 incredibly uniform 1 nanoliter droplets per minute.
100,000 droplets a minute.
Yeah. And here's the crucial bit. They carefully controlled the concentrations of cells and beads going in.
Why?
So that, based on Poisson statistics, the math governing random rare events.
Right, like how many raindrops hit a square foot per second.
Kind of like that, yeah. It ensured that most droplets would contain either 0 or 1 cell and 0 or 1 bead.
Ah, so you minimize empty droplets and droplets with, like, 2 cells.
Exactly. It makes the chance of getting exactly 1 cell and 1 bead in the same droplet pretty high, while keeping the doubles or triples really low. Super efficient pairing.
That sounds like this hyper-efficient biological assembly line working at a tiny scale.
Yeah.
Okay, so cell and bead meet in their private droplet. What happens inside? How does the info get, captured?
That's the STAMP workflow. STAMP stands for single-cell transcriptomes attached to microparticles.
Catchy.
Inside the droplet, the lysis buffer breaks the cell open, releasing all its messenger RNAs.
Okay.
Those mRNAs, with their poly-A tails, then find and stick to the oligo-DT part of the primers on the bead trapped in there with them.
So the message gets stuck to the bead's primer.
Right. And remember, that primer already has the cell barcode and the UMI. So the cell's message is now physically linked to its unique address and a molecule counter.
Clever. What happens after that? Do they process each droplet?
No, and that's the other piece of genius. After letting this happen for a bit, they break all the droplets open, pulling everything back together.
Wait, they mix it all up again. Doesn't that lose the single-cell information?
Not anymore, because the mRNA is physically attached to the bead carrying the cell's barcode.
Ah, the barcode preserves the origin.
Exactly. So now you have this pool of beads, the stamps, each one covered in mRNA specifically from one original cell. And then they do the expensive steps reverse transcription to make cDNA and PCR amplification, but they do it in bulk on the whole pool of beads together.
Wow. So they bypass the 1 reaction per cell bottleneck completely.
Totally. Huge savings in reagents, huge savings in time, but all the crucial cell of origin information is preserved on those beads.
Okay, so they've got this library of barcoded cDNA on beads. How do they read it all out and build that map, the expression matrix?
That's where the sequencing comes in, using standard paired-end Illumina sequencing.
Paired end, so reading from both ends of the DNA fragment.
Right. Read 1 gives you the cell barcode and the UMI. Tells you which cell, which molecule.
Okay.
Read 2 gives you the actual sequence from the 3-cut of the cDNA, which you map back to the genome to figure out which gene it was.
Got it. Barcode plus UMI from one read, gene identity from the other.
Exactly. Then software takes over, does error correction, counts the molecules using the UMIs, and automatically builds that gene-by-cell matrix, your digital expression matrix.
A map showing how much of each gene is turned on in thousands of individual cells.
Precisely.
This is where it just feels completely different. Because compared to what came before, like you said, this isn't just tweaking things, it's a quantum leap.
It really is.
Trading that full-length gene information, which older methods sometimes got.
Right, but very slowly and expensively.
For this massive, like over 100-fold jump in how many cells you could analyze.
At least 100-fold.
And the cost, going from maybe $10 a cell down to less than 10 cents.
Around 6.5 cents per cell, they estimated.
That's just, it makes it a whole different ballgame.
Yeah.
It takes it from something only specialized labs could dream of to something potentially many labs could actually do.
Absolutely. It democratized the whole field, really. But, with claims that big, they knew they had to back it up with some serious validation. Prove it worked.
All right, you got to trust the data. How did they do that?
One really clever approach was these species mixing experiments.
Human and mouse cells together.
Exactly. They took human HEK cells and mouse 3T3 cells, mixed them together, and ran them through the Drop-seq workflow.
Okay, what's the logic there?
Simple but effective. If the system works perfectly, a droplet with a human cell and a bead should only give you human RNA sequences attached to that bead's barcode. A mouse cell droplet, only mouse sequences.
And if you see a mix of human and mouse sequences linked to the same cell barcode?
That means something went wrong. Most likely you got two cells trapped in the same droplet, a doublet, or maybe a cell stuck to a bead from the wrong species, so that's less likely.
So what did they find? Did it work cleanly?
Remarkably well. They found that the individual stamps the beads after capture showed really high organism specificity. Very few mixed signals.
Good news for single-cell integrity.
Excellent news. It confirmed that mostly, yes, it was one cell paired with one bead, and it let them estimate the doublet rate. It varied a bit depending on how concentrated the cells were going in, but it ranged from as low as 0.36% up to about 11.3%.
How does that compare?
Much better than other methods available back then, which could easily have doublet rates from 11% up to 30%. So a significant improvement in purity.
Were there any impurities then?
They did acknowledge one source, which is common to pretty much all single-cell methods, a low level of background ambient RNA, probably from cells that got damaged during preparation, but it was minimal.
Okay, so it's specific one bead, one cell mostly.
Right.
What about sensitivity? Did it actually capture a good amount of the RNA from each cell, enough to get a decent picture?
Yeah, they looked hard at that too. For those human HEK cells, on average, they detected about 44,000 mRNA transcripts.
44,000 molecules per cell.
Yeah, and those came from about 6,700 different genes, on average, per cell.
That sounds pretty comprehensive.
It's a good snapshot. And to check the accuracy, they compared the Drop-seq expression levels to standard bulk RNA-seq data and also to other single-cell amplification methods. The correlation was really strong, like 0.90 to 0.94.
Meaning Drop-seq was measuring the right things in the right proportions.
Exactly. It accurately reflected the known gene expression patterns. And one more technical number, the capture rate, like what percentage of the original mRNA molecules actually got captured and sequenced. They calculated it to be around 12.8%.
That good.
For this kind of digital counting technology, yeah, that's considered pretty robust. You never capture 100%, but that's a solid efficiency.
Okay, so this level of validation, it's not just hand-waving. They really put it through its paces.
Oh, yeah?
You have to build that trust when you're introducing something so potentially disruptive. And it sounds like they proved it was robust, accurate, reliable, not just a cool idea. It actually worked.
It worked, yeah.
All right, so they built this incredible validated machine. Now, the exciting part, what could they actually do with it? What biological secrets could they uncover? This is where the science really happens, right?
Absolutely. And right away, they showed it could do more than just tell cell types apart. It could reveal different states within a population.
Seems like what the cells are doing.
Exactly. They looked at human and mouse cells that were just growing and dividing normally, asynchronously, no fancy treatments.
Okay.
Just by looking at the patterns of gene expression across thousands of individual cells, Drop-seq could figure out which phase of the cell cycle each cell was in, resting, growing, copying DNA, dividing, without needing chemicals to force them all into the same phase.
That's powerful. You see the natural dynamics.
And even better, by comparing gene expression across those inferred phases, they found new genes that seem to be regulated by the cell cycle. Genes like E2F7, PARPBP, SRP9, ATF4, stuff that hadn't really been linked strongly before.
Wow, so discovering potentially new players in a fundamental process, like the cell cycle, just by looking at lots of single cells.
Right. It showed the power wasn't just in cataloging cell types, but understanding their dynamic behavior.
Okay, that's a huge step
Yeah.
from who's there to what are they doing. And then the big test, tackling a really complex tissue. Where did they go for that?
They went for the mouse retina.
The retina. Why the retina?
It was a perfect test case. It's complex, has dozens of known cell types, photoreceptors, bipolar cells, amacrine cells, ganglion cells, gallia.
Lots of different jobs being done.
Exactly. But even with all that known complexity, many subtypes still didn't have good molecular markers. It was a great challenge for a new high-throughput method.
And the scale. How many cells did they profile?
This is where they really went for it. They profiled 44,808 mouse retinal cells.
44,000.
In one go.
In one early experiment, yeah. Audacious.
Wow. And the result? Did they build an atlas?
They did. They computationally clustered those cells based on their gene expression and identified 39 distinct populations. 39 types or subtypes?
Transcriptionally distinct populations, yeah. They could match most of these to the known major retinal cell classes, but they also found clear evidence for new candidate subtypes within those classes.
So refining the known map and adding new landmarks.
Exactly. And what was really striking was the impact of scale. As they analyzed more and more cells, the distinctions between closely related cell types became sharper, clearer. They could resolve finer differences and pull out rarer populations that would have been totally lost in smaller data sets or bulk analysis.
Like those amacrine cell subtypes you mentioned.
Precisely. Those were previously really hard to tease apart.
It really is like if you connect this to the bigger picture, going from a blurry satellite photo of a city to a clear street map, and then suddenly being able to zoom right into individual houses and see who's home and maybe even what they're doing, the granularity is amazing.
That's a great analogy. And they really did zoom in. They looked closely at those amacrine cells, the ones that process signals within the retina. They found 21 distinct clusters just within the amacrines.
21 flavors of amacrine cell.
Based on their RNA profiles, yeah. They could tell apart the major known groups, the ones using GABA or glycine or glutamate as neurotransmitters, and even these rarer ones called NGG amacrines. They could spot known types, like the ATI amacrines using the gene GJD2 as a marker, or the starburst amacrines using CHAT.
Okay. Confirming known markers.
But here's the discovery part. They nominated new potential marker genes for specific subpopulations that hadn't been well characterized, like a gene called MAF for one GABAergic group and PPP1R17 for an NG group.
So potential new ways to identify and study these specific cells.
Exactly. And they didn't just leave it as a computational prediction. They went back to the lab and used immunohistochemistry using antibodies to light up proteins in actual retinal tissue.
Right. Seeing is believing.
And they validated that these new markers really did label distinct populations of amacrine cells in the locations they expected. So directly expanding the known cell type map.
That's fantastic, connecting the sequencing data back to the tissue structure.
Yeah. And they even showed this power on really rare cells. They found a cluster of retinal ganglion cells, the ones that send signals to the brain. Even within that one cluster, they could zoom in further and identify the specific RGCs that express the light sensor melanopsin, OPN4, which are known to be involved in setting circadian rhythms. These make up less than 1% of all retinal cells.
Pulling out that needle from the haystack.
The sheer number of cells they profiled made that possible. It gave them the statistical power to see these rare types and their unique gene signatures.
Okay, so zooming out again, what does this all mean for the field? You said it wasn't just an incremental tweak, but a paradigm shift.
How so?
It really boils down to scalability and accessibility. Being able to make 10,000 single-cell libraries in, 12 hours?
Incredible speed. For about 6.5 cents a cell. That just blew the doors open. Single-cell transcriptomics suddenly became feasible for way more labs, for way bigger questions. It wasn't just for the elite few anymore.
It democratized the technology, like you said.
Totally. And it's also fascinating how it happened. This wasn't one lone genius. It was this amazing cross-disciplinary team effort.
Right. You mentioned computational biologists, engineers, physicists.
Yeah, from Broad, Harvard, MIT. They were all working together, apparently, with very few intellectual property constraints holding them back early on.
That probably helped speed things up.
Hugely. They talked about how prototypes for the microfluidic chips could go from a design on the computer to being tested in the lab in just days. Feedback from sequencing runs would immediately inform tweaks to the bead chemistry, like week by week. Really rapid iteration.
That sounds like a very dynamic environment.
Yeah.
And the people involved, you mentioned their risk appetite, running nearly 45,000 cells on one of the first experiments. That takes guts.
It really does. People like Evan Macosko, who developed the bead strategy, Anindita Basu engineering the droplet chip, Rahul Satija building the early analysis tools, which became the basis for Surat, a standard tool now.
Oh, wow.
Stephen McCarroll pushing for that massive scale and getting the sequencing resources, and Aviv Regev, who really framed the whole idea of building cell atlases as this grand challenge. Their combined vision and boldness was key.
A real confluence of talent and ambition. And this wasn't just about making catalogs of cell types, was it? The potential goes way beyond that.
Oh, absolutely. The future applications are what get really exciting. Now you can start linking those big genetic studies, the ones that find genes associated with disease risk.
GWS studies and things like that.
Yeah. You can finally ask, okay, in which specific cell types is this risk gene actually active, and what does it do there? Not just in the whole tissue average.
Connecting genetics to cell-specific function.
Precisely. And you can combine Drop-seq with experiments. Treat cells with drugs, introduce mutations, infect them with pathogens, and then use Drop-seq to see how each different cell type responds.
A multidimensional view of biology in action?
Exactly. It gives you this incredibly detailed cell-by-cell readout of complex processes. That opens up huge possibilities for understanding disease mechanisms and finding much more targeted therapies.
It really makes you wonder if a gene's function isn't just about the gene itself, but about the whole cellular neighborhood it lives in, its context.
Right.
What other hidden relationships, what undiscovered cell states or rare but crucial cell populations are just waiting out there now that we have tools like Drop-seq to find them? It feels like we've just been given the keys to a whole new layer of biology.
It really does feel that way.
Well, that's a wrap on this deep dive into how a truly clever mix of beads, droplets, and barcodes smashed the single-cell ceiling and opened up this microscopic world. It's just a fantastic example of how pushing technology lets us ask entirely new scientific questions.
Indeed. The ability to routinely profile thousands, now even millions, of cells has fundamentally changed cell biology, developmental biology, neuroscience, immunology, cancer research, you name it. It's a testament to how understanding and applying core principles can just accelerate everything.
And remember, the great thing about science is there's always more to uncover. What stood out to you in this story?
Oh, sure.
What new questions does this kind of technology make you think about? Keep digging, keep exploring, and we'll see you next time on The Deep Dive.